Welcome to the Explainer. Okay, let's dive into this incredibly elegant, systemic solution to how your body powers itself. Today, we are tracking energy all the way from the literal food on your plate right down to the exact microscopic cellular level. If you've ever wondered how your morning toast physically translates into brain power, you're in exactly the right place.
Here is our blueprint for today. We'll be moving through fueling your brain, looking at simple sugars, breaking down those building and breaking reactions, checking out complex chains in storage, testing for carbohydrates in the lab, and we'll wrap it all up with some absolutely crucial exam tips.
All right, let's kick things off with Section 1, fueling your brain right from the source.
Did you know that the bread you had for breakfast could be the literal reason you aced your math test? It's true. Carbohydrates are your body's primary energy source. They are your absolute go-to premium fuel, powering everything from the physical contraction of your muscles when you're moving around, all the way to those intense, glowing neural pathways firing away in your brain.
And the sheer output is just massive. For every single 1 gram block of carbohydrates you consume, you get an explosive 17 kilojoules of energy. When we're talking about biological efficiency, understanding this exact 17 kilojoule metric is essential. It's kind of like checking the exact octane rating of the fuel you're pumping into your car.
So, what exactly is this fuel made of down at the atomic level? Well, carbohydrates are organic molecules built strictly from carbon, hydrogen, and oxygen. But here's the absolute key to remember. There is an exact 2-to-1 ratio of hydrogen to oxygen. You definitely want to lock that in, because that 2-to-1 proportion perfectly mirrors the structure of a standard water molecule. Think about it. Carbon plus water, literally carbohydrate.
Moving right along to Section 2, the simple sugars are biological building blocks.
Before we can construct massive, complex biological structures, we really need to understand our base ingredients. The general formula here is CH2O taken N times, where that N ranges from 3 to 7. These are your monosaccharides, things like glucose and fructose, and they are your absolute base building blocks. You can think of them as indivisible single Lego bricks. You just cannot mechanically break them down any further into simpler sugars.
Now here's the complete categorical breakdown you're going to want to perfectly memorize. Starting from our base, we have those single Lego bricks, the monosaccharides, glucose, fructose, and galactose. Now snap 2 of those together and you step up to disaccharides, sucrose, lactose, and maltose. Finally, when you string tons of them together into massive, complex chains, you get polysaccharides, which include starch, glycogen, and cellulose.
All right, let's jump into Section 3, building and breaking reactions. Let's really look at the molecular mechanics here.
You will absolutely be tested on this mechanical process, so let's track exactly how this works. This is what we call a condensation reaction. First, 2 individual monosaccharide blocks meet. Then, an entire water molecule, H2O, is physically extracted and pulled right out of the structure. The removal of that water literally forces the 2 blocks to lock together, building a larger disaccharide. So just remember, condensation equals building by removing water.
On the flip side, hydrolysis is the exact opposite mechanical process. Instead of water leaving, a water molecule is aggressively driven back into the bond. Think about the word itself. Hydro means water, and lysis means to split. By forcefully adding water back into the equation, you snap those 2 Lego blocks right back apart into their original monosaccharide base units.
Which brings us to Section 4, complex chains and storage, basically the cell's energy vault.
Okay, so what happens when you link hundreds or even thousands of these monosaccharides together through those condensation reactions? You create polysaccharides. Imagine a massive winding string of pearls, where every single pearl is a standalone glucose unit. Because these structures are so incredibly large, they are completely insoluble. They will not dissolve in water. And crucially, unlike those simple sugars, polysaccharides are absolutely not sweet. This actually makes them the perfect, stable, non-reactive vault for long-term energy storage inside a cell.
What's really fascinating is how different branches of biology handle this massive energy storage. Animals, which includes us, take any excess glucose and tuck it away as a polysaccharide called glycogen. We pack this tightly into our liver and muscle tissues for quick deployment when we need it. Plants, however, have a dual strategy. They use starch purely to store energy, just like a potato does, but they use an entirely different polysaccharide called cellulose to build their actual physical structural components, like rigid stems and cell walls.
Moving to Section 5, testing for carbohydrates using the Benedict's test.
So how do we actually prove these simple reducing sugars exist in a lab setting? We use the Benedict's test, which is a strict 3-step process. First, you drop Benedict's solution straight into your sample tube. Second, you absolutely must apply active heat. This reaction is not going to happen cold. Third, you watch for the physical color shift. If that sample morphs from clear blue, passes through green and yellow, and finally hits an undeniable brick red color, boom! You have explicitly proven that reducing sugars are present.
Finally, Section 6. Exam tips and a rapid recap so you can master your test.
A massive exam tip here is to heavily drill your understanding of those molecular mechanics. Seriously, don't be the student who gets these mixed up. Condensation means water is removed to build. Hydrolysis means water is added to break apart. Secondly, never fall for the super common trap of assuming all carbohydrates taste sweet. Just remember that massive string of pearls, huge polysaccharides like starch and cellulose, have literally zero sweetness.
All right, let's hit our rapid-fire summary to lock in the core data points you must know. Remember your atomic elements and that exact 2-to-1 hydrogen-to-oxygen ratio. Memorize that 17 kilojoules per gram energy metric. Clearly separate the simple, single monosaccharide blocks from those massive polysaccharide chains. And lastly, know the exact steps and that blue to brick red color change for the Benedict's test completely inside out.
I really recommend you practice these concepts. Sketch out those mechanical flow charts for condensation and hydrolysis on your own, and make sure you can really visualize how those molecules build and break. But most importantly, knowing exactly how these bonds store and release 17 kilojoules of energy per gram, how are you going to physically fuel your own brain for your next exam? Make sure you sit down with the right knowledge and, hey, for sure, the right breakfast before test day. Thanks for joining this explainer, and keep learning.